Okay, I am starting my class today, and in today's class, I will start your genetics part, and that is, I'll start right from Mendel's Law. I have already sent you people. I have already sent you people a video link of a class I had earlier taken, and so you people, I hope, will go through those videos and then kindly listen to what I say here. So, I let me share my whiteboard. So please do inform me if you are able to see my whiteboard. Are you able to see my whiteboard? Huh? Yes?
Yes, sir.
Okay, fine. So let's start with Mendel's law. Mendel was a monk, he was a father, an abbot in the monastery of Brunn. Currently, it's now in Czechoslovakia, or rather in Czech. So he did some crosses with pea plants. He did some crosses with pea plants, and as a result of those crosses, roughly in the year he did this crosses, roughly 1865 to 1869. He did all these crosses, and as a result of these crosses, which were basically statistical analysis done after cross pollination. So what he got from that, he gave three laws. First was the principle of dominance. In this, what he did was, when he crossed a tall variety when he crossed a tall variety of pea plant with a dwarf variety of pea plant. Just hold on, somebody is entering. So he crossed with a dwarf variety of pea plant. What he got was, all the plants were tall. So at this point, let me explain what an allele is. Allele is basically a contrasting form of a gene in the same loci of the chromosome. So if I draw a chromosome, this one is obtained from your mother, this one is obtained from your father. So allele over here, this is, say, capital A, and this is small a. So A and A, they are alleles of each other. Maybe say this A and A, they control a particular phenotype, say height, height of plant. So when he did, so this is an allele. So what Mendel did was, when he crossed TT into TT, these were the tall plants and these were the dwarf plants. When he crossed them in the this was the parental generation. He got heterozygous tall plants, the first filial or the F1 generation. He got tall plants. So the tall character overpowered or dominated the dwarf character, and this he called the principle of dominance. The next thing what he did, the next principle he had was the principle of segregation, where these tall plants and dwarf plants, they were crossed, and he self these plants, the F1 were selfed and crossed among themselves, and he got it in the ratio of what he did was, he got tall plants, he got 3 tall plants, and at the same time the dwarf character re-emerged. The dwarf character re-emerged. So when the dwarf character re-emerged, that meant that this TT into TT, when this F1 self cross was being done, the gametes separated or segregated independently. And because of this segregation, these characters were you had 3 tall and the re-emergence of the dwarf character. At the re-emergence of dwarf character. So the dwarf characters re-emerge. So this was the principle of segregation. And this type of a cross where only one character was considered this is known as a monohybrid cross. The one character considered was plant height, and it had two forms. One was tall, the other was dwarf. Capital T, small t. T and T are alleles of each other. In the next cross, Mendel performed a dihybrid cross. On the basis which he gave the third principle known as principle of independent principle of independent assortment. And what is this principle of independent assortment? He used two characters. One was yellow, round, the other was green, wrinkled. So pea pods
স্যার কিছু শোনা যাচ্ছে না, আপনি বোধহয় মিউট করে ফেলেছেন।
Okay, now am I audible?
Yes, sir.
Okay. Yes, sir. Okay. So one was yellow round and one was green wrinkled. So the pea were yellow, round and green wrinkled. Yellow round and green wrinkled. And say the characters were capital G, this was for yellow, and capital, one second, this I have written wrong here. Capital W for round. So this was for yellow color, and this was for round shape. This was crossed with green wrinkled. So one thing was sure, that yellow was dominant over green and round was dominant over wrinkled. So this was what he got from the monohybrid cross, law of dominance. Law of dominance. So when this particular cross was done in the F1 generation, as expected, what he got was he got the heterozygotes. And they were selfed. So result was, if we write it down as GW let me use one single color for simplification. So he had gametes forming. He had GW, G small w, G capital W. These were the gametes which were formed. So when he these were crossed, what he got was G capital W. He got G capital W small w. He got G W. Then if you write got it like this, G capital W small w, capital G small w. Then you have capital G small g, capital W small w. Then you have capital G small w. Here capital G small g, capital W. Here you have capital G small g, capital W small w. Then small g, capital W. Then small g, capital W, small w. Capital G, capital W, small w. Capital G, small w, small w. Then you have small g, capital W, small w, then you have W's, this finally. Now if you look at this particular thing, this was yellow round, this was also yellow round, this was also yellow round, this was also yellow round, this was yellow round. But here small w there. A new assortment came, a yellow wrinkled. It is a new assortment, a new combination, new assortment or new combination which came for the first time. This is again yellow round, and again here you have yellow wrinkled, this is yellow round, and again for a new combination comes the green round. Green was normally wrinkled, another new assortment or combination which has taken place, new combination. This is also the same thing, green round, then you have this yellow round again, yellow wrinkled again, green round again, and green wrinkled again. So if you see the combinations, yellow round, yellow wrinkled, green round, green wrinkled, you get it in the ratio of 1, 2, 3, 4. 9 is to 3 is to 3 is to 1. 3 is to 3 is to 1. And this is a dihybrid cross. And this laid the principle of independent assortment. Now I would go to the next part of my talk, which is extension of Mendelism. Extension of Mendelism. And now what is the extension of Mendelism say? We had talked about alleles being the contrasting form of a particular gene. Now there is a term known as incomplete dominance. In the flower snapdragon, thing was found when red flowers were crossed with white flowers. Instead of getting red flowers, what they got was pink flowers. Mixture of white and red. So if you say the phenotype, look at the phenotype, this is the genotype and amount of gene product. In the red flower, capital W, the genotype, two alleles, two copies of red flower genes are present, and so the gene product is 2x. In the pink flower, one capital W and you had a one small w. Result product is 1x, one time gene product. So this was red, 2 times gene product red, 1 time gene product pink, and this was the no gene product of red, 0. You had absence of red pigment, white flower. And this phenomenon is known as incomplete dominance. Now we will go to the next phenomenon, which is known as codominance. Humans have a blood group known as blood group L, blood group M, and blood group N, and it had 2 alleles, blood group L and blood group N. These are the 2 alleles, 2 blood groups, 2 type of special type of blood groups. M and M. This is the MN system of classification. This is the MN system of classification. Now what you find over here is that if we write this is the genotype, the blood type, that is L antigen present, what type of antigen is present, and if you see if the genotype is LM, the blood type is M. You have M antigen present. If you have LN, blood type is N. It is. But if it is LN, both M and N antigen are present. So it's MN blood group. So both M and N are present. M is not dominant. M is equal to N. So they are codominant. The next concept which we will be studying is multiple alleles. Our ABO blood group is a good example. So you have more than 2 forms of a gene is present. So there you have several alleles. In the same locus. It can be IA, it can be IB, it can be IO. In this case, A and B are codominant. A is dominant over O and B is dominant over O. So this is known as multiple alleles. Next we come across a phenomenon known as epistasis. This happens when there are two or more genes, they are influencing a trait. It happens in this case, an allele of either A or B, it can be small a or small b, they can have an overriding effect on the phenotype. This phenomenon is known as epistasis. In the sweet pea, Lathyrus odoratus, it happened that when one variety of white variety flower 1 was crossed with another variety flower 2, suddenly you had purple flowers coming up. This was very strange, white variety 1. So Bete Sun and Puneet, they suggested that this color was controlled by two genes. One was C and one was P. The pigmentation was because of this purple color was because of a pigment known as anthocyanin. This anthocyanin was the result for this pigmentation. And for this anthocyanin, you needed to have a dominant C as well as a dominant P. So in this part, it can be a capital C, small c. Here it can be capital P and small p. It could be anything which could fall for purple color pigmentation for anthocyanin biosynthesis. So in this cross, the two varieties of white which were crossed were capital C, small p into, this was white 1, white 2, small c, capital P. In both of this was a recessive, this was recessive. As a result, the flowers were white because you needed at least one copy of capital C and capital P for the color, for anthocyanin biosynthesis, and the color of the flower to be purple. So the result was, when the hybrid came, it was capital C, capital P. It was a mixture because of independent assortment. And this was a purple flowered because anthocyanin was started to be produced again. Now when they were selfed, the following results came. Instead of 3:1 or 9:3:3:1 or whatever, they got a ratio of 9:7 of purple is to white. CP, 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 small c, small p. Now I draw this. And now if I make this this Bete Sun and Puneet square, it came. This type of analysis was done by Bete Sun and Puneet itself. Some people call Bete Sun and Puneet. Some people call Bete Sun and Puneet. Capital C, capital C, capital P, capital P. This is like this. The same thing, like independent assortment, this thing starts to take place. Capital C, capital C, capital P, capital P, capital C, capital C, small p, small p, capital C, small c, capital P, small p. Capital C, capital small c, small p, small p. Capital P, capital P. So in this you could find this to be purple, 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 since both of them have to be purple, 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 purple. This is purple. 1 2 3 4 5 6 7 8 9. So 9 purple is to the remaining 7 white, and this phenomenon is known as epistasis. So with this, I have already sent the notes, my notes uh, in the Microsoft Teams. So you could go through my notes. There are certain small minor topics also additional in that, and with that you can actually, I think that would be sufficient for your exams. So let's get back. So do you people have got any questions? Questions? Questions আছে কোনো? Aritri?
না স্যার।
Okay. So no questions today. Fine. Then I end my class. I will tell when the next class is. Okay. Thank you very much.
থ্যাংক ইউ স্যার।
Hmm. You will get
Thank
the
you, sir.
you will get the recording also automatically in Teams. I don't have to send it to you also.